Welcome back, BioMonsters. Today we're still going to be talking about genetics, but we're going to be focusing on when heredity follows different roles. So today we're going to be talking about things that Mendel had no idea about because he didn't have the science to support the math. So let's go ahead and dive in. To get started, let's just quickly review some things that Mendel taught us. So he said that alleles, and remember alleles are the same thing as genes, only he didn't know about genes at the time, come in two forms. And those two forms are either dominant or they are recessive. And you guys should remember that if it's dominant, we're going to be representing that by a capital letter. And if it's recessive, instead, we're going to use a lowercase letter. He also said that each one of these alleles that come in two forms have to be paired. So our alleles will be paired, and when they are paired, they can take on one of three different pairings. So if we are talking about something that is homozygous, meaning the same, homozygous dominant, so we know we're going to be using capital letter, capital letter. We could also be homozygous, so again, still the same, recessive, and that means lowercase letter, or we can be heterozygous, and when we're heterozygous, that means different or other, so we're going to have two different letters. So we're going to have a capital letter and also a lowercase letter. Let's talk about some simple recessive disorders. So let's just talk about the cause really quickly. So again, we're talking about recessive disorders. So its cause, it's due to a recessive allele, or we can say a gene, okay? And the possible genotypes for actually having this recessive disorder, we'll think back to what we just talked about. The only way to see a recessive phenotype is if it's homozygous recessive. So the only possible genotype for having a recessive disorder is to be, once again, homozygous recessive. And what does that look like? That looks like two lowercase letters. The last thing that we need to know about simple recessive disorders is that there are things called carriers. So remember, this is the only way to have the disease, right? So the only way to actually have the disorder is to be lowercase, lowercase. So we know that it's also possible to be homozygous dominant. And if you're homozygous dominant, so a capital letter, something that you need to know is that you are unaffected. So you don't have the disease, but it's also impossible for you to pass it on to the next generation. However, there are things called carriers. Now, carriers always have the same genotype. So the genotype for a carrier will always be heterozygous. And let's talk about why. For a heterozygote, you're always going to have a capital and also a lowercase. So we'll use T's again, so we'll use a capital T and a lowercase t. The reason why we call this genotype a carrier is because, look, they are carrying this little t and they can potentially give it to the next generation, even though they themselves do not have the phenotype for the disorder because this capital letter is keeping them safe or keeping them healthy. Well, the first simple recessive disorder that we're going to talk about is something called cystic fibrosis. Cystic fibrosis, also referred to as CF, is unfortunately a very common genetic disorder, and in fact it is the most common genetic disorder found in white Americans in this country. So if we were going to talk about what the statistics are, it's actually 1 in 20 white Americans carry, so that means they are heterozygous for this recessive allele. It doesn't mean that they have it, it just means that they have one of the lowercase letters and they can potentially give it to the next generation. Because 1 in 20 are carriers or heterozygous, 1 in 2,000 children will actually inherit the disease. And to inherit the disorder, you have to be homozygous recessive, because this is a recessive trait or recessive genetic disorders. So what you need to know about CF, you need to know some general things about the disorder and what it causes. CF, or cystic fibrosis, generally affects the lungs and the upper digestive tract, specifically the respiratory system, and it causes a thick buildup of mucus. And unfortunately, when you have a thick buildup of mucus, it prevents you from participating in gas exchange. And now that we know what cystic fibrosis is, let's actually look at a word problem, because this is what you're going to see on your test and also on your quiz. So here's our word problem at the very top. we want you to cross a male who is unaffected, so they do not have the disorder, with a female who is a carrier for cystic fibrosis. So I'm hammering out the problem right now, trying to figure out what all the key information is. So if I know that this person is unaffected, it means that they do not have CF, right? But then we also have a carrier, and so what we know about a carrier is that they can pass it to their offspring. So let's go ahead and write that just to keep that fresh in our minds. Now, luckily, I've already given you the genotypes for each one of these parents. So I have parent number 1 who's unaffected and parent number 2 who is a carrier. So I'm going to go ahead and write down my parents over here. So parent number 1 is going to be big F, big F. Parent number 2 is going to be big F, little f. So we have that information. I don't have to make my key necessarily because I already have my letters here, but I'm going to go ahead and make one anyway just to help keep things straight in my brain. So I know capital F is unaffected, so I'm going to write that. Lowercase f, you have CF, right? So let's go ahead and fill in our Punnett square then. Remember, we always put our parents along the top and the left-hand side. It doesn't matter where you put parent 1 or 2. I'm going to go ahead and put parent 1 across the top. I'm going to put a capital F, that's one allele that they have, and here's another allele that they have over the other box on the Punnett square. And I'm going to put my other parent who is heterozygous or a carrier along the side. Now remember the way that we fill in Punnett squares, it's exactly the same. Whatever's on the sides coming in, whatever's on top's coming in, and that's going to be true for all 4 boxes. So I'm going to go ahead and fill it in. For my first box, I know I'm going to be big F, big F. For my second box, big F again, and then big F. For my third box, big F, little f. And again, for my last one, big F, little f. And to make it easier for me to understand, I'm going to go ahead and fill in what each one of these phenotypes is actually going to be. So in my first box, if I'm big F, I know that I am unaffected. So I'm going to go ahead and write that so it'll be easier to answer my questions down the road. And obviously this is the same thing, so I'm going to put that there as well. And then along the bottom, I'm big F, little f, so I know that this is a carrier, so they do not have the disease personally, but it's possible for these children in the future to pass on the disease to their offspring, and it's true for both of those boxes. So now that we have that filled in, let's go ahead and look at our word problems. The first one says, what is the probability? So I'm looking for a percentage of producing a carrier child. And remember, my carrier is a heterozygous genotype. So if I look, I have 2 boxes that are heterozygous, and each box represents 25%. So I have 25 plus 25, so if I have 2 quarters, that makes 50 cents, or in this case, 50%. So if these 2 parents had children, 50% of their offspring potentially could be carriers for the disease. Let's look at the next question. What is the probability of producing a child who actually has CF? Well, remember, if we think about what we just talked about, the only way to have CF is to be homozygous recessive, because it is a recessive disease. If I go through all of my boxes, I'll notice that I have no children or no boxes that are lowercase, lowercase. So that means the probability of these parents having a child who actually has the disease is 0%. All right, we've come to your first stop and jot. Go ahead and pause the video, and when you think you have the right answer, call your teacher over to check for correctness. Our next recessive disorder that we're going to talk about is a really difficult word to pronounce. It's a disease called fetal ketonuria. We usually just refer to it as PKU. This is still a recessive disorder, so we want you to think about what we talked about for CF, because the same rules still apply. So again, it's a recessive disorder that prevents the production of a very important enzyme. And remember, in order for it to be recessive, it has to be lowercase, lowercase to actually have the disorder. Well, what does it mean if we don't have this one enzyme? We've talked about enzymes before. We know that they speed up chemical reactions. So I guess to really understand what PKU is and how significant it is if it actually occurs in an individual is to figure out what this enzyme actually does. So if we look down here, it says this enzyme converts something called phenylalanine into a different amino acid. And the reason why we need to convert phenylalanine is because if it builds up in the body, it can actually cause severe damage to the central nervous system. And unfortunately, if it continues to happen, it can actually cause death. Here's our first one. So again, we're going to hammer out the problem. We have a carrier male, and remember, to be a carrier means that you are heterozygous, so you yourself do not have the disorder, but you are holding one lowercase letter, so it's possible for you to give it to the next generation, with a woman who has the disease. So she has PKU, and the only way to have it is to be homozygous recessive. So let's go ahead and figure out who our parents are. So I'm going to write that up here. Parent number 1, I already know, is a carrier male, so it's going to be heterozygous, big A, lowercase a. And the woman in this particular word problem actually has a disorder, and the only way to have it is to be homozygous recessive, so little a, little a. I'm also going to fill out a key for myself so I don't get confused. Remember that PKU is a recessive disorder, so if you have a capital letter, it means that you are unaffected, but if you have a lowercase letter, it means you have PKU, unfortunately. So let's go ahead and see if we can fill in our Punnett square. We're going to do the same thing that we did last time. We're going to put one parent here and one parent along the side. I'm going to put parent 1 across the top, big A, little a, and parent 2 along the side, little a, little a. It doesn't matter in which place you put parent 1 or parent 2. Just to remind you, the way that we fill in our Punnett squares, again, whatever's on the left-hand side comes into the box, and whatever's on top comes into the box. And the way that we always do things, we put the capital letter first. So let's see what we're going to get. In box 1, I'm going to get a big A, little a. In the second box across the top, I'm going to get a little a, little a. In my next box, I'm going to get a big A, little a. And then in my last box, a little a, little a. And just to make sure that I don't confuse anything, I'm going to go ahead and write down what these phenotypes are. If I'm a big A, little a, again, I know that I don't have it, but I'm also not unaffected. Instead, I am a carrier. So I'm going to write that down. And I'm going to look at this other box that looks exactly the same, and I'm going to write the same thing. This box also represents a carrier. My last 2 boxes are exactly the same. They're homozygous recessive. So that means that this box represents an individual with PKU, as does this one, unfortunately. So now let's go back and see if we can answer some of these questions. What is the probability? Again, that means percentage of producing a carrier child. So we have to ask ourselves again, what is a carrier? A carrier is always going to be heterozygous, so we don't get confused. So all I'm going to do is I'm going to quickly look through my boxes. I see that I have a carrier here and a carrier here. So 2 boxes represent carriers, and each box represents 25%. So if I add them up, I know that the possibility of these 2 parents having a carrier child is going to be 50%. My last question is, what is the probability? So again, I'm talking about a percentage of producing a child who actually has PKU. Well, remember, the only way to have a recessive disorder is to be homozygous recessive, which means lowercase, lowercase. If I quickly scan my Punnett square, I notice that I have a lowercase, lowercase here, lowercase, lowercase here. So again, each box represents 25%. So unfortunately, these 2 parents have a 50% chance of having a child who also has PKU. All right, here's your next stop and jot. Go ahead and pause the video, and when you think you have the right answer, call your teacher over to check it for correctness. What we're going to talk about now is one example of dominant heredity. And for dominant heredity, remember, dominant means a capital allele. So think about what you know about capital alleles. They're strong, they have all the power, and a lowercase allele cannot mask what's on the capital letter. So let's go ahead and then answer what it's caused by. It's caused by a dominant allele. And remember, the word allele is just another way of talking about a gene, which means that it has to be a capital letter. So it's caused by a capital letter. The possible genotypes now to actually have a dominant a dominant genetic disorder, it's going to be one of two types. So you can either be homozygous dominant, which means that you have two capital letters, so it would look like a capital D, or you could also be heterozygous, which is new because with a recessive disorder, heterozygotes do not have the disease. But if you're heterozygous, you're going to be a capital letter, lowercase letter, and remember what we said, this guy right here is really strong, so this guy can't mask what's on that capital D, so you will still have the disorder. The next question is, do we have carriers? For simple dominant heredity, there's no such thing as a carrier, so no carriers exist. And the reason why no carriers exist is because if you are heterozygous, you automatically have the disease. Dominant disorder that you are responsible for knowing is a disorder that's horrible. It's a horrible disease called Huntington's disease. Huntington's disease is a deadly genetic disorder and it's caused by a rare dominant allele. So the only way to have it is to have at least one capital letter. It results in the breakdown of certain areas of the brain, so basically holes develop in your brain if you have Huntington's disease. The onset of the disease usually occurs between the ages of 30 and 50. Here's our word problem. If we look here, it says that we want to cross a male who is heterozygous for Huntington's disease. Remember, if you're heterozygote, you have the disease if it's dominant because you have at least one capital letter. With a person who is unaffected. And if you're unaffected, it means you have to be homozygous recessive. So I have my two parents here. So parent number one is going to be big H, little h, so heterozygous, and they have it. Parent number two is unaffected, so they're going to be little h because this is a dominant disease. Let's go ahead and write in our key. Remember that if you are a capital letter, you have the disorder because it's dominant. And if you're lowercase, you are unaffected. Let's write that just so that we don't get confused. Now that we have our parents and we've hammered out our problem, let's go ahead and see if we can populate our Punnett square. So I'm going to put parent number one across the top. You're still going to use your Punnett square in the exact same way that you used it for a recessive disease. You're just going to read it a little differently. And my next parent is unaffected, so it's going to be lowercase h, lowercase h. And just a reminder again, remember whatever's on the left-hand side comes in, whatever's on the top comes in. So let's quickly see if we can populate our Punnett square. In this first box, I'm going to have a big H, little h. My next one, little h, then big H, little h, and then lastly, little h. And just like we did before, I always like to write down what the phenotype will actually be. It makes it easier to tackle the second half of the word problem. If I'm big H, little h, do I have it or do I not have it? Remember, it's a dominant disease. As long as you have at least one capital letter, you have it. So we're going to say that this person is affected. They have the disease, or the person represented by this Punnett square, or this quadrant. Looking at my next one, I'm lowercase. Remember, it's a dominant disease, so if you have no capital letters, you are unaffected. So this person does not have it. This person is exactly the same, or this quadrant is exactly the same, so I'm going to write unaffected. And here we have a capital letter and a lowercase letter, and again, as long as you have a capital letter, you have the disease. So I'm going to go ahead and write affected. Now that we have that information, let's see if we can go ahead and answer the question. The first question asks, what is the probability, and again, probability means percentage, of producing an unaffected child? The only way to be unaffected when we're talking about a dominant disorder is to be homozygous recessive. So we're going to quickly look through our Punnett square. Do we see any homozygous recessive genotypes? I do. I look here. This is homozygous recessive. This is also, so I have two quadrants. Each one represents 25%, so it's going to be a total of 50%. So these two parents have the option, if they have children, there's a 50% likelihood that their children will be unaffected, will not have the disease. If I move on to the next question, it asks again, what is the probability, so I'm looking for a percentage, of producing a child who actually has the disease. Remember, there are two ways to have a dominant disorder. You can either be capital H, so homozygous dominant, or you can be heterozygous, big H, little h, because as long as you have a capital letter, you have the disease. So if I look through again quickly my Punnett square, I notice I have two quadrants that are affected, and each one represents 25%, so I know that this is also going to be 50%. Stop and jot. Go ahead and pause the video and answer the questions on your stop and jot on your day sheet, and when you think you have the right answer, call your teacher over to check for correctness. When we're talking about genetics that doesn't work the way Mendel predicted, we're talking about something called non-Mendelian or alternative inheritance patterns. So this means we're not simply going to be talking about something that's just dominant or recessive. But before we talk about the new stuff, let's quickly recap. So remember that when we talk about Mendelian inheritance deals with simple dominant and recessive alleles. So here's an example. Dominant alleles will always be expressed and they will always hide the recessive allele. So that means that the capital letter is stronger than the lowercase letter. So here's an example. If I were to cross a parent who had dimples, and dimples are a dominant trait, so they're capital, that's their genotype, with a parent who has no dimples, so they're homozygous recessive, we would get a child who has dimples who is a heterozygote. Let's put that into a Punnett square so we can actually see how that transmission happens. So I'm going to go ahead and put parent 1 across the top, parent 2 along the side, and I'm going to populate my Punnett square just like I've always done. And when I do so, I notice that 100% of my children will actually have dimples. So that's what we see right here. This is pretty predictable. Mendel was able to use his pea plants to understand that dominant alleles will always mask recessive alleles. But unfortunately, the way that peas transmit genetic information is not the way that we transmit all of our genetic information. So it doesn't account for a lot of the other traits that we're seeing that either get blended and or you see the expression of both alleles. So let's talk about what that actually looks like. So the first new term that we're going to talk about is something called incomplete dominance. This is an important vocab word, and here is the definition. So an incomplete dominance, the name actually explains how this transmission pattern actually works. So here, both alleles, remember my alleles are my letters, are expressed to create a blended phenotype. And by blended, we mean a mixed phenotype. So in incomplete dominance, you're going to see a new third phenotype. That's unusual because when we talked about Mendelian genetics, you either have dimples or you don't. So there are only two phenotypes possible. You're either tall or you're short if you're a pea plant. So again, there's only two phenotypes possible. In incomplete dominance, the way that you automatically know that we're dealing with incomplete dominance is that you're looking for a third phenotype. Let me show you what I mean by that. So when we look at our example, snapdragons, which are a type of flower, come in 3 flower colors. They could either be red, and if they're red, they're capital R, or they can be white, and if they're white, they're lowercase. But if you see a heterozygote, the heterozygote has a brand new phenotype, and that brand new phenotype is going to be pink. So let's talk about what that looks like in a cross. So I'm still going to set up my Punnett square. It's still a monohybrid cross. We're just dealing with one trait, the trait of flower color. I'm going to put my one parent up at the top, big R, that's representing my red snapdragon. I'm going to put my white snapdragon along the side, little r. And when I do a cross, you'll notice that all of my Punnett squares are going to become heterozygotes. Now, if this were simple Mendelian genetics, we would say that all of these guys simply look like the capital R. But this is incomplete dominance. And in incomplete dominance, all of these guys, because they're heterozygotes, will now be pink because these guys do not one doesn't have the ability to mask the other, so they blend together. So they're incompletely dominant. They're just going to blend together and form a third phenotype. So all of these guys now are going to be pink instead. And if you look at our picture, this is a good example. So here's my red snapdragon, which is big R, my white snapdragon, which is little r, and then all of my babies from that cross are going to be big R, which is my new phenotype. So again, the key here, you're looking for a new phenotype, a third phenotype, which is basically just a mixture or a blending of the other two homozygotes that are possible within that particular trait. Next, stop and jot, go ahead and pause the video, and when you think you have the right answer, call your instructor over to check for correctness. Our next new form of inheritance is going to be something called codominance. And in codominance, we're going to focus on this word co. When you have a co-pilot or a co-president, you guys know that those two individuals have equal power. So they're not going to give in to the other individual. So unlike incomplete dominance, where those two homozygotes simply blend together, in codominance, they're not going to blend together. They both want to be equally as strong. So instead of seeing a blending, you're going to see a combination of both in the heterozygote. So what's the definition? Here it is right here. Both alleles are expressed equally, but they are also expressed separately. So another way of saying it, you will see both phenotypes in the heterozygote. So let's talk about an example because that can be pretty tricky. So here's my example. Cows have the possibility of coming in for this particular example. They can be red, or they can have a red coat, which means that they are big R, or they can have a white coat, which means that they are capital W, capital W. And notice that I'm using two different letters here, and both of them are capital. So they're going to be equally as strong, and they're not going to give in to one another. But it is also possible to get a cow that is called a roan or has a roan coat, which is a mixture of both red and white hairs. And the only time you would see that is if you have a heterozygote, and that heterozygote is going to be capital R, capital W. Let's go ahead and do a cross and see what this actually looks like. So I'm going to draw on my Punnett square. This is still a monohybrid cross because we're only dealing with one trait, which is fur color. I'm going to put one parent across the top, big R, another parent along the side, capital W, and when I fill in my Punnett square, all of my babies end up being heterozygotes. And if they're all heterozygous and they express both fur colors, we know that it has to be codominant because we don't see a third phenotype. Here's your next stop and jot. Go ahead and pause the video, and when you think you have the right answer on your day sheet, call your instructor over to check for correctness. In multiple alleles, we're going to see three different possible alleles that can make up a specific phenotype or make up a genotype that gives rise to a phenotype. So what do we mean by multiple alleles? Well, the name pretty much tells you. It means that you have more than two versions of a gene. So our example that we're going to use in class is going to be blood type. Now, some of you might already know this, but when we talk about blood type, there are basically three possible alleles. You can either have a capital A, you can have a capital B, or you can have an O. Sometimes these guys are represented this way instead. There'll be a capital I with a superscript A, there'll be a capital I with a superscript B, or there'll be a lowercase I. So it depends on what the word problem looks like, but either way you need to make sure that you know we're talking about the same thing here. What you'll notice when I set it up this way is that if I use a lowercase I, which is representative of the O allele, that this is the one that is recessive. And that's important. These two are both going to be dominant. And so we're going to see some interesting behaviors when we actually do some blood typing. So let's go ahead and look at our possible blood types. It's possible to have a blood type which is type A, and if you are type A, here are your possible genotypes. You could be big A, or big A, O. It could also be represented this way. You could be capital I superscript A, capital I superscript A, or you could be capital I superscript A lowercase I, which again means the same thing. For blood type B, there are two possibilities again. You could be big B, or big B, big O. It's also represented this way. You could be capital I superscript B, capital I superscript B, or you can be capital I superscript B and then a lowercase I. Next possible genotype is blood type AB. Now for blood type AB, there's only one possible genotype, and that genotype is capital A, capital B, or we can write it this way, big I superscript A, big I superscript B. And what you guys should immediately notice here is that for this particular blood type, this also looks like something that we've seen before because it expresses both alleles. So we see both phenotypes, not a blending. So this is also an example of codominance. codominant. So it's not only an example of multiple alleles, but blood typing could also be used for this particular phenotype as codominance. Our last possible blood type is blood type O, and for blood type O, the possible genotype is OO, or we could say it's lowercase I because blood type O is recessive. So let's go ahead and do a cross so you can see what this actually looks like. So I'm going to set up my Punnett square. You can do the same thing on your paper. I am going to cross a parent. So listen carefully. Parent number 1 is going to be heterozygous for type B blood, and parent number 2 is going to be heterozygous for type A. So what does that look like? So if I'm heterozygous, I know I have 2 different alleles. So I'm going to do capital I with a superscript B, lowercase I, or you can write it big B, O. And then for parent number 2, it's still a heterozygote, so we have 2 different alleles. It could be capital I superscript A with a lowercase I, or it could be A, O. I'm going to use my I with the superscript to put it in my Punnett square because I think it's easier to read. So I'm going to put parent number 1 here, capital I with a superscript B and a lowercase I, and parent number 2, capital I with a superscript A and a lowercase I here on the side. We're still going to follow the same rules that we always do for our Punnett squares. Whatever's on the side comes in, whatever's on the top comes in, and I'm going to keep everything in alphabetical order. So in my first box, I'm going to bring in my capital IA, capital IB. In my next box, capital I superscript A with a lowercase I. In my next box, capital I superscript B with a lowercase I. And then in my last box, 2 lowercase i's. So if I'm looking at this, what are the possible phenotypes for my babies? Well, it is possible if we look at my first quadrant, if I'm capital I superscript A, capital I superscript B, I know that I'm blood type AB. That's my codominant phenotype. For my next one, I'm capital I superscript A and a lowercase I. And remember, this guy is not strong, so it can't mask what's here, so this is going to be blood type A. In my next box, if I look, I again have a capital I superscript B with a lowercase I. That lowercase I is recessive, so it can't mask my B. So this is going to be blood type B. And then in my very last box, I have 2 lowercase i's, which represent type O blood. So I'm going to go ahead and write O. All right, we've come to your next stop and jot. Go ahead and pause the video, see if you can answer the questions on your day sheet. And when you are done, call your teacher over to check for correctness. The last thing that we need to discuss is something called polygenic inheritance. And we've talked about this prefix poly before. Remember, poly means many. And we want you to remember that because in polygenic inheritance, we're going to see many, many, many different types of phenotypes. And the reason why is because we're going to see a spectrum, and that spectrum is due to the fact that more than one gene controls a particular trait. So you guys have probably figured out by now, for instance, if you look at humans, what the inheritance patterns that we've talked about so far still doesn't explain the broad spectrum or range of phenotypes that we see for a particular trait. The example that we're going to use today is skin pigment. So if we look at the coloration of skin just within our own species for humans, you'll notice that there's no such thing as a white person and a black person. That when we look at different types of people, even if they share a similar background, there are variations in their skin pigmentation. Well, the Mendelian genetics that we talked about, as well as the alternative forms of inheritance that we've talked about so far, don't explain how we got all of these extra phenotypes. So the only thing that does explain this is something called polygenic inheritance, and remember, poly means many. So here, we're going to see the interaction of many genes. So we're not just talking about one gene. We're talking about 6, 7, 8, 10 different genes that all control that particular phenotype. And because so many genes are responsible for the phenotype that you will actually see for that particular trait, you will see a spectrum. And another word for spectrum is range. All right, we've gotten to your last stop and jot. Go ahead and pause the video, answer the questions in your day sheet, and when you think you have the right answer, call your teacher over to check for correctness.